Okay, good afternoon, everyone, and thank you so much for joining us again on this Friday afternoon. Today, we have the pleasure of listening to Professor Gerard du Preez, who is an Associate Professor in Agricultural Sciences at Northwest University. For those of you who don't know, Prof. du Preez specializes in nematology and soil ecology, and his research is focused on sustainable and climate-smart agriculture, primarily investigating soil health and ecosystem functionality across diverse farming systems. So he's very qualified to speak to us today about soil health and the biodiversity that supports it. Just before I hand over, though, please remember to add your comments or questions in the chat box. We won't be taking live questions, but please add them to the chat box and we'll get to them at the end of the session. And a reminder for those who are participating in the CropLife CPD program, the assessment is available on the CPD system in category 2 of the online modules. So thank you so much for your time to speak to us today, Prof. du Preez. Over to you.
Hi, Ariza, and good afternoon to everyone that's attending. Thanks for the opportunity. It's always a pleasure to speak about soil health and especially the biological component of soil health. I've been involved in the agricultural sector for about 10 years. Actually, I always called my previous life, in terms of my academic career, I was studying nematology, but actually in caves. But somewhere along the line, I realized that I probably want a job one day. So I decided to switch over to pivot to the agricultural sector from the beginning of 2015, also at the start of my PhD. And I've never looked back. It's been a great journey till date, and I'm really looking forward to meeting new people and also establishing new collaborations. So I'm going to switch off my camera just to make sure that there's no bandwidth issues, but I'm sure everyone can see my screen. All right, so as Ariza said, we'll be talking about soil health today, and specifically the role that biodiversity plays in supporting it. And just the screen that you see here, in fact, on the title page, it's a photo from one of the trials that we're involved with. And I always like to include this on the title page because this trial is part of what we call the FIP program that's managed by Asset Research and Dr. Hendrik Smith and his colleagues. It's a trial that's implemented on farmer fields. And I think that just tells the story that the research that we're doing, we're really focusing on the questions that the farmers are asking. And so whenever we deal with soil health and whenever we deal with the biological component or the biodiversity that's supported, we always think it's very important to really look at the benefits that farmers can expect. So that's also something that we'll address during this presentation. Right, so looking at the definition of soil health, the one that you see on the screen is one that I think you've all read or heard about before. But I always like to emphasize a few things. Reading this, you can see that there's a key focus on the ability of the living ecosystem to function. And that's also a central focus of today's talk. And I'm going to explain to you exactly why ecosystem functioning, or the ability of the ecosystem to function, is such an important concept when we're dealing with soil health. And that ultimately allows the concept of soil health to sustain plants, animals, and humans. So traditionally, when we think about soil health, we have these three main components: the chemical component, the physical component, as well as the biological component. And we have different, we call soil health assessment frameworks that include different parameters, basically a subset of parameters representing each of these components. And they use different scoring and weighting systems to ultimately create something called a soil health score. And that's what we use to try and get some meaning from all the analyses that we do. One example is the SMAF framework, the one that you see on the screen here. One that's probably more commonly known in South Africa is the soil health tool, or actually better known as the Haney analysis. And then also the comprehensive assessment of soil health, which is a Cornell framework. So these are probably the three main frameworks currently used in the industry. And like I said, each of them have a different subset of parameters that are integrated, weighted, and scored into something that calculates a soil health score. The reality is, though, that there's a problem with the biological component. Biology is not simple. Biology is complex. Biology is not easy to measure. It requires expertise to analyze. It requires expertise to interpret. So this is something that we have to acknowledge, is that the chemical and physical components are typically very well represented in these frameworks. But because the biology is costly and laborious and also often time sensitive, they don't receive the same focus as the physical and chemical components in these soil health assessment frameworks. So that's something we need to take note of when we think about the biology in soil health, and we need to place more emphasis on this. So of course, a lot of this has to do with biodiversity. So I wanted to start talking about biodiversity in the general sense of what we understand biodiversity to be. So if I ask you what is biodiversity, a lot of you will say probably the number of species in a given area, and that's accurate. But actually biodiversity is the number of species in a given area as well as its distribution, so how evenly distributed do the species occur in that environment, so linking also to its abundance. But we also know that, for example, in 1 teaspoon of healthy soils, we can have, as the picture shows, several thousand different species or taxa, hundreds of meters of fungal hyphae, or 1 billion bacterial cells. So that is typically something what we associate with the concept of biodiversity. We also think of biodiversity as something that's abundant in different systems. Here we can see an example of a huge amount of insects that we can find in natural ecosystems, but also in agricultural systems. And we know that insects, for example, are incredibly important. Just think about, for example, of pollinators. We know that food security is intricately linked to the ability of insects to pollinate our crops. And to give you a little bit of reading material, I always advise to go and read up or Google this report by the FAO, State of Knowledge of Soil Biodiversity. It was published in 2020. It's free. You can just search for it. It's really a big report, but it contains beautiful images, but also very, very useful information, specifically on the relevance of biodiversity in our natural world. All right, so I want to break this down a little bit more. I now explained to you that we get a different number of species, and we also look at how abundant and evenly distributed they are. But there's not just species diversity, there's also other types that it's important to take note of. The one is genetic diversity. So within a species, so within one population of a species, we'll have variance or variation. Just think about humans, for example. We don't look the same, we look different. That is genetic diversity within the species. Now, species diversity I've already spoken about, but then we also have something else that I think is quite fascinating, and in my mind is becoming more and more important also in the agricultural context or in agricultural landscapes. And that is ecological diversity. So ecology, the word ecology is from the Greek word oikos, which means house. So ecology is really the study of organisms or living organisms in their habitat or in their housing environment, if we want to call it that. And if you think about the natural world, there's a lot of variation in the natural world. And although we tend to think of agricultural landscapes as more simple, the truth is that there can also be high variability between different farms. Even on a farm, there could be high variability in soil types or rainfall or the topography and general landscape characteristics. Now that's very important because the environment selects for what species occur in that given place. And that all links up to ecological diversity. Now there's a fourth type of biodiversity that I really like to emphasize, and this is the direction where a lot of the research that we do is heading towards, and it's called functional diversity. Now, I have to admit, functional diversity is something that's sometimes difficult to explain because there's been over the years many different definitions of what functional diversity is or what ecosystem functions are. But to boil it down into simple terms, we now agree that functional diversity basically explain the range of bundled processes that are performed by living organisms in a given environment or in a given ecological setting. So we know that organisms in the soil perform specific processes. And we're going to talk about that a little bit more in the next few slides. But we need to think about that there are different processes and different organisms performing different processes. So when we talk about functional diversity in a given environment, we're really trying to imagine what are all the bundled processes that are performed by the community of organisms that inhabit that specific ecological setting. So to try and explain this a little bit more and link the organisms more with processes, I'm using this schematic illustration from a paper published in 2022. You can see that the biology links up with the soil processes and that they ultimately deliver what we call soil functions. And we're going to, through the next few slides, break this down a little bit more. So let's start with the processes. Now, these you've probably heard about before, and I'm going to give you one or two additional examples. But bioturbation, exudation, macropore formation, predation, microbial grazing, and so forth. So the one example I want to look at is bioturbation. I find it is actually quite fascinating. So this is a YouTube video that you can find online from the authors I cited. What's really great about this, it really perfectly illustrates the process of bioturbation. So here you have two mesocosm experiments. On the left-hand side, you'll see that all the living organisms have been removed. That mesocosm has been sterilized. On the right-hand side, you can see the activity, that living component really doing its work. It's breaking down that organic material. It's pulling that down and incorporating that into the soil. And although this was at a higher speed, this entire process was only happening over the course of a few weeks. So this is bioturbation, and this is one of the processes that the soil ecosystem fulfills. And just by looking at the image that you have on the screen now, you can clearly see the difference in organic matter breakdown and incorporation into the soil if we have that living component versus when it's absent. So another example is predation or even microbial grazing. So what you see here on the screen is C. elegans. It's a nematode, and it's feeding on an agar plate on E. coli bacteria. So you can imagine that nematodes have the ability to regulate microbial communities, to either stimulate or reduce their populations because it feeds on them. And then another example would be this predator. So this is a Mononchidae nematode actually feeding on another nematode. It's quite a gruesome video, actually. But you can imagine this other nematode it's feeding on, maybe it's a pest nematode. So nematodes actually have the ability to regulate other nematode populations, and they even have the ability to regulate pests and diseases in agricultural settings, but also in nature. This happens in nature as well. Now, I'm including this, and I'm using nematodes as an example because, as you heard during the introduction, I am a nematologist by training, and nematodes, of course, fascinate me. And just maybe an interesting fact, nematodes are, in fact, the most abundant multicellular organism on Earth. And there was a very famous nematologist who unfortunately passed away, but his name was Nathan Kopp. And he used to describe the number of nematodes in quite an interesting way, and I'll quickly relay that same message to you. He used to say that if you can imagine all the valleys, the mountains, the rivers on Earth, but suddenly you take them away, all the material, all the matter on Earth you take away, except for the nematodes, you will still be able to recognize those mountains, valleys, and rivers, simply because there are so many nematodes present in these habitats. And that's truly the case. Nematodes occur almost in all environments, even extreme environments. And they often have a bad rep because, as you will know, especially in the crop protection industry, nematodes can cause a lot of economic damage. Thinking about root knot nematodes, lesion nematodes. But the truth of the matter is that by far the majority of them are actually beneficial and deliver very important ecosystem processes that ultimately contribute to the functioning of that ecosystem. So to just show this schematically, I've included this image that's actually included in a chapter that we'll publish soon. What you see here on the left-hand side are the different trophic groups. So these are nematode trophic groups. So, I mean, they're very broad classifications. Within each of these trophic groups or feeding groups, there are hundreds to thousands of species. So the links that you see here to the next level, the processes, are really only representing the broader links. So you can imagine that there are actually hundreds of thousands of nematode species that classify within one of those trophic groups. But then what's important is that these nematodes fulfill specific processes in the soil. So here we've got microbial grazing I spoke about earlier, as well as predation. We also have parasitism, so EPNs, or entomopathogenic nematodes, actually have the ability to parasitize insects, for example. Then we also have resistance and defense mechanisms. Microbial dispersion is another important process. So these processes that nematodes fulfill ultimately then contribute to what we call subfunctions and ultimately functions. Now, we're not going to talk about this in depth, but you can see that nematodes are actually contributing to the four main ecosystem functions, which you can see here on the right-hand side. But what I want to emphasize, just for the sake of this presentation, is the disease suppression and pest control ability, as well as the plant health promotion ability, because that ultimately contributes to disease and pest regulation. So, like I mentioned, nematodes can graze on microbial communities. So nematodes have the ability to suppress pathogens. They have the ability to feed on nematode pests. But they also have the ability to promote plant health. How do they do that? Well, it's actually one of the areas of science that's developing, and as you will know, that more research is needed. But we know that nematodes have the ability to disperse beneficial organisms. And by dispersing beneficial organisms, they can stimulate the interaction between, for example, the microbial community and the plant host. And that ultimately can lead to a positive effect on plant health. So that ultimately contributes to what we call the disease and pest regulation function. So in summary, this is not simple, it's complicated. What you see here is from that same paper from 2022, and it's the state of knowledge at that time in 2022 when this paper was published. And it's complicated. There's so many links, and it's difficult to make sense of it all. But in summary, you'll see that we have the darker brown nodes. They represent the different organisms. We've got the beige or lighter colored nodes. They represent all the processes. So all the organisms, the organism groups are linked to these processes, and then they ultimately link to the blocks that you see that represent those subfunctions, and to summarize it all, ultimately contribute to the four main ecosystem functions. So when you read these four main ecosystem functions, nutrient cycling, carbon and climate regulation, water regulation and purification, disease and pest regulation, without having to say much about it, I'm sure we can all agree that these functions are really important, and they can play a very important role in promoting soil health and to the benefit of farmers. So when we think about how we manage our agricultural systems, we need to start to think also about the ability of the ecosystem to perform, to function within that management context, and to link that with specific benefits that the producer or the farmer can receive, or expect at least. So to break this down even a little bit more and to bring it more into the context of agriculture, I always like to use this infographic. So I shared the link down at the bottom, but you can just Google SARE soil health infographic on Google, and you'll probably find this is the first return. What makes it very useful, it's like I said, it's an interactive tool. So if you go online, you can click on these different icons. It provides you with more information. People who are familiar with soil health, I'm sure there's something that you can learn from this as well, but it's probably most valuable for people who don't know much about soil health. So I always say to people, share this with people that you want to learn something about soil health. It really is a very useful tool that I use in class as well. The point that I want to make here that's quite important is what we've said now is that management practices, in this case here, we've got compost, tillage, the use of, for example, cover crops, livestock integration, rotation systems. These management practices all have a very important influence on the ability of the soil organisms to function. So remember we spoke about earlier about ecological diversity. So organisms are sensitive to management practices. Some are more sensitive than others. So what we do in the soil, how we till the soil, what products we apply, for example, all of that has an influence on the ability of the soil ecosystem and the organisms within that ecosystem to function, to fulfill those important processes. And that directly links to the benefits that farmers and producers can expect. And these benefits, or we call soil health benefits, these can truly link to a monetary value. So when we talk about agricultural landscapes, and this is probably one area of research that really needs more focus, is linking the soil health benefits to a monetary value, so that we can better understand, and farmers can better understand, by implementing a certain agricultural system with set practices, we actually have a greater potential to receive certain benefits, which can ultimately help us save costs, and while doing that, also reduce the impact on the environment. So what I also want to do during this presentation is not just talk theory. I also want to share with you some results from a few of the projects we've undertaken in South Africa. So it's actually just 2 projects for this presentation. So the first is a case study. So this is a study that we did on 2 farms. The one was close to Reitz, the other was close to Vrede. So we did it on the farmers' fields, basically just going out there and over 2 intervals, sampling the fields without us as the researchers having any intervention in the actual management practice. So it's really a snapshot of what the condition of the soils were like at the time of sampling, given the farmers' management context. And also very importantly, and what I'll get to a little bit later, given the specific environmental context as well. So the question we were asking, or the question we wanted to ask was, or to answer was, what influence does the different conservation agriculture context have on soil ecosystem status? So in this case, we were only focusing on conservation agriculture, and many of you will probably know conservation agriculture is broadly defined by a few principles. So reduced physical disturbance, or even no tillage in some cases. Crop diversification, so rotation, but also sometimes the integration of cover crops. And then also soil cover being very important in the context of conservation agriculture. So we wanted to understand what was the influence of the implementation of conservation agriculture on these 2 farms, on the soil ecosystem, and not just soil health, but really focusing on the biological component of soil health. Because as I said during that introduction of this presentation, historically, we haven't given much thought about biology, but that needs to change. So what I'm showing here is the first set of results from Vrede. Now, this is a multivariate analysis. So what you can imagine this is basically a 2-dimensional plot, an ordination plot of the different fields that we sampled, but also plotted on the different parameters that we measured. So when you look at the left-hand side, you can see that there are different symbols. Now those symbols represent the 3 conservation agriculture fields that we sampled at Vrede. The 1 conventional agriculture field presented by the red diamonds that we also sampled. And then we also included a pasture system. So we included the conventional and the pasture systems as reference systems, so we can compare that against the conservation agricultural system. And then the different ecological indices that we measured, you can see here, are written in black font, and we'll deal with a few of them in a moment. So what's important to understand about these kind of graphs is that things that are grouped together were similar at the time of sampling. Things that are further apart or opposite each other, plotted opposite each other, are dissimilar or may present a negative correlation. So let's take, for example, the pasture system here on the left-hand side of the plot. You can clearly see the pasture system plotted separately from the agricultural system. So that's a conservation and the conventional system. But it also showed quite a strong correlation to the maturity index, the soil respiration values, as well as organic matter content. So the maturity index is actually a nematode-based index, and it measures what the name actually suggests, the maturity of the ecosystem. So it basically tells us how healthy the soil ecosystem is. And because it's so closely associated with the pasture system, given also the agricultural systems on this plot, we know that in the pasture system, the soil ecosystem was healthier. And we also see that by having higher soil respiration rates, which is basically a measure of microbial activity, as well as organic matter. But this is not surprising. If you think about it, agricultural systems are disturbed quite frequently in different ways, physical disturbance, chemical disturbance, whereas pasture systems are often grazed only once or twice a season and then left alone, often with no physical or chemical input. So that has the ability to then restore and their ecological balance and basically produce a healthier and functioning soil ecosystem. But nonetheless, looking at the agricultural systems, we also notice something interesting. So from this initial graph, you might think that all the agricultural systems just plotted together. But actually, there was a very clear separation between the conventional system and then the conservation agriculture systems. And we tested this statistically. So I'm not showing the stats here, so you're going to just have to trust me on this. But the conventional system plotted lower in terms of soil ecosystem health, in terms of organic matter content, in terms of soil respiration rates, compared against the conservation agriculture systems. So all three conservation agriculture systems scored higher. Now, when we do these kind of studies looking at the biological component, we should not neglect the physical and chemical components. Although, as I've mentioned now a few times, we really need to emphasize the biological component. It doesn't mean that we should completely neglect the chemical physical component, because those measurements, the physical chemical properties, help us to understand what's happening in, or what performance is happening in the ecosystem itself. So in this case, we plotted the physical chemical properties that explained most of the variation we saw in the biological properties, also on this graph. But you can see where my arrow is pointing that there's not a lot that's being explained by the physical chemical properties. The major distribution on this graph was between the pasture and the agricultural systems, and that was on the horizontal axis. But what you see here on the physical chemical properties is that they really only explained some of the vertical distribution, which in the case of the biological properties was not that meaningful. Now I want to show you the results of Vrede, and I'll go through this a little bit quicker. What's interesting here is that it showed us the opposite picture of what we saw at Vrede. So here we've got conservation agriculture system 1, we've got conservation agriculture system 3, and then we have the pasture system down here. So you can clearly see clustering happening. So the conservation agriculture system 2, the light blue colored diamonds, are also forming a cluster. So you can clearly see the different clusters forming, and there's no clear separation between, for example, the pasture system or the conservation agriculture system. There's also no clear separation between the conventional and pasture system or the conventional and conservation agriculture systems. So this is quite interesting. So it became, well, we became aware, we learned that basically the management context, the management practices on this farm did not have such a pronounced effect, not to the same extent as what we observed in Vrede. But when we looked at the physical chemical properties, now there was a different story to tell. So when you look at the properties listed here, yes, there are some nutrient level considerations, but the main separation between these ecological properties and the clusters that we see on the left-hand side was as a result of soil texture in this specific case. So soil texture really had a big influence. So you can see that the conservation agriculture system 1, the blue one, had much higher sand content, whereas the other systems had higher clay content, and especially the conservation 3 system at the highest clay content. So that was an interesting finding, but also a very important one. So the key findings was that the management context influenced soil health at Vrede, and specifically also that conservation agriculture scored higher than the conventional system. At Reitz, though, we saw something different. where the environmental context really influenced soil health, and the main driver in this case being soil texture. So I also want to quickly talk about that second case study I mentioned earlier. So in this case, we selected different ecotopes. So an ecotope is basically an area, a given area with a similar climate and potentially similar soil types. So you can see here on the bottom left image, I encircled the sampling sites. Some of them were close to Potchefstroom, so in the Viljoenskroon area. A few of them were the Bergville area, but then also in Mpumalanga. So we really wanted to understand what is the potential influence of variability in ecotopes. So basically, different agricultural systems scattered across different ecotopes. And in this case, we focused on conventional systems. And when I say conventional, I just mean business as usual. Your conservation agriculture system, which includes the practices that I previously mentioned. But then we also included regenerative agriculture. And in this study, the separation between conservation and regenerative agriculture was simply that with regenerative agriculture, the farmers had to implement cover crops as part of the rotation and also livestock integration on those cover crops. So that was the only separation between conservation and regenerative agriculture. Again, as with the previous case study, we wanted to get a better understanding how the soil ecosystem status was influenced by these different farming systems, but also across different or multiple ecotopes. So here again, we have that maturity index. I'm going to try and activate my pointer quickly. All right, so here we have on the left-hand side the maturity index. And as you'll see, we have the different ecotopes listed: Ascent, Bergville, Reitz, Standerton, Viljoenskroon, Vrede. And on the right-hand side, the plot gives a separation between the different agricultural systems. So CA, conservation agriculture, CV, conventional agriculture, and RA, regenerative agriculture. So what's interesting here, and what you can see as well very clearly on this first diagram or plot, is that there was quite substantial variance in the maturity index between the different ecotopes. So again, the maturity index is a measure of soil ecosystem health, and it varied quite substantially between the ecotopes. So the maturity index only really ranges from 1 to 5. So the fact that you have Ascent plotted closer to 2 in terms of the median, and Vrede much higher to 2.6, 2.7, that is quite a big difference for this particular index. However, when we look at the agricultural systems, there were no significant difference. So the statistics also supported this. So there were statistical significant differences between the ecotopes, but none between the agricultural systems. Just to show another index, which is also quite useful, it's the enrichment index. So it's an index also measured by studying the nematode community. And it tells us basically what is the level of nutrients available to the soil ecosystem and indirectly to the crops in a given environment. Again, you can see high variability between the ecotopes, but almost no variability between the agricultural systems. So this again was an interesting finding. And I included this particular analysis as well, because here we're integrating some of the physical chemical components as well. So on the left-hand side, again, we have very clear separation as a result of those ecotopes. So here, for example, is Ascent, and we've got a cluster here of Reitz, Standerton, Viljoenskroon, and Vrede, and then Bergville down here in the bottom. And you can see the main properties contributing towards the separation. So there's definitely some physical chemical properties, pulling, for example, in this case, Ascent, but then also biological properties here, the maturity index. When we look at the agricultural systems on the right-hand side, there's lots of overlap between the clusters, which basically means that there's minimal separation between the agricultural systems. So the key findings from this case study was that soil ecosystem status did differ between the ecotopes, and it was definitely influenced by the physical chemical properties, but that there were minimal differentiation between the agricultural systems. Now, I should say, because some people might be quite upset with this finding, because they might quote me in saying that, no, but now you're saying that conservation or regenerative agriculture has no influence or no effect. That's not what I'm saying. What I'm saying is that given the context of this study, this is what we're seeing. But what we will do, because the study is ongoing, it's still preliminary results, we have our third year of sampling next year. And after the third year, we'll do the same analyses, but we'll take it a step further. We'll also do in-depth analysis of each ecotope, similar to what we saw during that first case study of Vrede and Reitz, to really understand what were the main drivers within each ecotope, not just across all the ecotopes. So that's something that we will work on within the next year or so, and we will publish that result. So in part, my conclusion, coming back to this soil health infographic, this link between management practices and the functions that the soil organisms perform, and then ultimately the benefits that we can expect, this is context dependent. Context dependent in terms of the management context, so what management practices are implemented, how frequently are they implemented, to what extent are they implemented. And then, of course, also the environmental context, linking back to that original explanation I gave about ecological diversity. It is really important to consider the environment under which we're operating. We know that one set of rules do not apply everywhere. So, and that goes for conservation and regenerative agriculture as well. So on-farm testing and verification is indeed very, very important. And to that end, I've listed here a few practical implications which might be of use to some. So what we've learned is that it's really important to properly characterize your soils, in terms of the type, the texture, also the depth. We also advocate that you understand or learn more about your natural context. Because if we think about it, if we are trying to farm in partnership with nature, we're trying to farm in a way that we promote nature to give us benefits to perform those functions, we have to understand how they actually function, given the environment in which we're operating. Now, I know that the natural context is not the same as agricultural setting, but it certainly can give you a better understanding of what species occur in that environment, naturally occur in that environment. And what do they actually do in that environment? So by learning more and more about your farm and where your farmlands are located, we can make informed decisions. So when I say natural context on a farm, we usually include some kind of reference system. You saw from the case study that we included a pasture system. Now, pasture is not 100% natural context, but it's a lot better than a intensely managed agricultural farmland or crop production system. But often we have parcels of land that are not intensively managed, and those can also serve as a good reference. It's important then, once we have all of these properties, to consider farm heterogeneity, because we know that within a given field, there can be large differences in soil type, for example, and that can and most certainly will almost have an influence on how your soil ecosystems perform. You really have to think about what it is that you want to achieve from a soil health perspective. You as a farmer, or you as an advisor to a farmer, what is it that the farmer wants to achieve? If it's to build carbon because he wants to sell his carbon credits, then there's certain practices, and for example, cover crops that you can plant that can help that or boost that. Do you want to cycle nutrients? Well, there's certain practices that boost that as well. What about pest and disease regulation? Same goes. So it's really important to think about what it is that you want to achieve. We have to design those agricultural systems linked to those outcomes. And then very important in my mind is a monitoring plan. We need data. We need good data. Data that we can trust, that was sourced or generated using an acceptable method that we know will be accurate. Because we can only make informed decisions and we can only adapt once we have a good monitoring plan in place. On-farm trials is something I always advocate for. Like I mentioned during my title slide, we have been involved in a number of farmer innovation programs and running trials on farmer fields. Exactly for the reason I've stated before, because we need to understand how certain systems function within a farmer's given environmental context. And we need to do that in more places. But what's very important is that it needs to be correctly designed, and the implementation needs to be done correctly. Because otherwise, unfortunately, we cannot use the data, we cannot trust the data. And we need the data. We need accurate data, again, to make informed decisions. Lastly, but not the least, I always say, try and link up with study groups, try and link up with experts, people who's been doing it for a while, who's been practicing conservation, regenerative agriculture, or whatever agricultural system you're implementing. Maybe it's organic agriculture, or maybe it's conventional agriculture with just a little stronger focus on biologicals, whatever the case might be, whatever your outcomes might be. Implement or rope in the experts to help you. And when you send your analyses to different labs, or when you have to decide on a lab, make sure that it's a reputable lab. It's very important. And I always advocate for looking for labs that are accredited. But I also want to advise against switching labs frequently. Yes, sometimes you have to switch for whatever reason, but there will always be variance between laboratories because there are humans involved, and there's always some form of human error involved in these kind of analyses. So try and find a good lab and try and stick with it as long as possible. I wanted to give this warning, and it's not because I'm cynical or want to be critical or anything, but there's a lot of evidence out there showing the potential effect of pesticides and herbicides on soil ecosystems. This is one review paper, which means that they looked at all the published literature and they basically made a summary assessments of all the results that were available. And the paper does show that basically across most, if not all of the classes of pesticides, it certainly has an influence on soil biology. I'm not saying for one moment that we should cut synthetics out, just leave them alone. I'm saying that we should always be aware of what we do and try and limit the application if possible. So basically advocating for the judicious use and not overuse of synthetics. What's now becoming more important or relevant is the use of biologicals. And where I'm based in Northwestern University, we're implementing a new research farm. We're calling it the NW Agri Hub of Excellence. One of the main questions we'll be looking at is the potential role of biologicals and how we can integrate biologicals in agricultural systems. And that's not just in regenerative systems, that's also in conventional systems. How can we implement biologicals in your business as usual systems and hopefully start reducing our dependence on synthetics? I'd like to acknowledge all the researchers involved, all the farmers, all the students, and then of course all the funders that allow us to do this research. Thank you. That's my story.
Excellent, Professor. Thank you so much for a very insightful and very valuable presentation. I'm very sure our members learned quite a bit with this. I see we do have a question in the chat box. Are you able to view the question and perhaps provide an answer, please, Prof? You are on mute.
Sorry, I showed my face, but I didn't unmute myself. Okay, so the title of the FAO document, let me pull it up quickly. What I'll do is I'll just post the link to the report in the chat, and then everybody should have access to it.
Excellent, thank you so much. I don't think we have any other questions. We did ask and remind people a bit earlier. So with that, I think we can conclude today's session. Thank you again for your time. Really some insightful presentations. Appreciate it very much.
No, it's a pleasure. Hope to chat soon again.
Great. Thanks so much. Thanks everyone for attending, and have a great Friday and weekend.